Have you ever stopped to admire the beauty of a flower? Flowers are the reproductive parts of angiosperms. There are two major groups, dicots and monocots. Dicots have leaves with branching veins. Monocots have leaves with parallel veins. The journey of a plant begins with a seed, a tiny package containing all the genetic information needed to grow into a mature plant. When we examine the seeds of dicots and monocots, we find a fundamental difference. Dicot seeds have two cotyledons, which are essentially seed leaves, that provide nourishment to the young plant as it emerges from the soil. Imagine splitting open a peanut seed and finding two halves. Those are the cotyledons. Monocot seeds, in contrast, have only one cotyledon. Think of a corn kernel. It has a single cotyledon that absorbs nutrients from the endosperm, a starchy tissue within the seed that provides food for the developing embryo. This difference in the number of cotyledons is a defining characteristic of dicots and monocots and has important implications for their early development. We've already touched upon the difference in leaf venation between dicots and monocots, but let's delve a little deeper into why this difference is significant. The veins in a leaf are like the plant's circulatory system, carrying water and nutrients from the roots to the leaves and sugars produced during photosynthesis back down to the rest of the plant. The net-like venation of dicots allows for a more intricate and efficient distribution of these vital substances. The parallel venation of monocots, while seemingly simpler, is well suited to their growth habit. Many monocots, like grasses, have long, narrow leaves that grow from their base. This type of growth, called intercalary growth, allows them to withstand grazing and mowing, as they can quickly regrow lost tissue. Roots. Beneath the surface, dicots and monocots also differ in their root systems. Dicots typically have a taproot system, characterized by a single, dominant root that grows straight down into the soil. Think of the deep roots of a dandelion or a carrot. These are examples of taproots. Monocots, on the other hand, typically have a fibrous root system, consisting of numerous, thin roots that spread out horizontally near the surface of the soil. This network of roots forms a dense mat that helps prevent soil erosion and efficiently absorbs water and nutrients from the upper layers of the soil. The roots of grasses and many other monocots exemplify this fibrous root system. Supporting the plant body. The stem of a plant provides support, holding the leaves up to the sunlight and transporting water and nutrients between the roots and the leaves. Inside the stem, we find vascular bundles, which are like the plant's plumbing system, containing xylem and phloem tissues that transport water and nutrients throughout the plant. In dicots, the vascular bundles are arranged in a ring around the central pith, which is a soft, spongy tissue that stores water and nutrients. If you were to cut a cross-section of a sunflower stem, you would see this ring of vascular bundles clearly. Monocots have a different arrangement of vascular bundles. Instead of being arranged in a ring, their vascular bundles are scattered throughout the stem. This scattered arrangement provides flexibility and strength, allowing monocots to bend in the wind without breaking. The pith in monocots is less distinct than in dicots and may not be easily visible in a cross-section.